Life defies simplicity, and this is one property of it that we have yet to discuss in our search for an adequate definition of life. Complexity, the organization that characterizes every life form, from amoebas to antelopes. Life is highly organized, and we even call living things organisms and talk about their parts as organs and organelles. Why should life be so organized? And is there an organization that we can see in single-celled beings that corresponds to that of more complex ones, like us? These are some of the questions that science seeks to answer. And by seeking the answers, we learn new and wonderful things about life and how it came to be. We've talked about the molecules that support and make up life. So now it's time to move up one level of organization and consider the building blocks of living organisms, the cells. Cells are the smallest, organized, self-contained living systems. They can be entire organisms by themselves, or only one of billions of cells making up a complex human being. Let's consider this for a moment. Every living thing is made up of cells, whether we're talking about mold or squid or humans. Why? Because we all came from the same starting point, and despite the long and convoluted path of evolution, have retained many features of our tiny one-celled ancestors. By some measures, 5 to 7% of our DNA is the same as that in bacteria. We share 15% of the genetic material found in mustard grass and more than 80% of zebrafish DNA. We truly are all connected to all of the other living things on Earth. As we've learned, cells are tiny bags full of organic compounds, but just as those compounds are intricately organized chains of carbon and its atomic buddies, cells have their own structure and key bits. Broadly speaking, there are two types of cells in living things, prokaryotes and eukaryotes. Prokaryotic comes from the Greek and means before kernel, where the kernel refers to the nucleus of the cell. Prokaryotic cells carry their genetic material in a region that is not enclosed by a membrane like the nucleus in a eukaryotic cell. Eukaryotic means true kernel and underscores the fact that eukaryotic cells protect their genetic material with a special double membrane wall that surrounds the nucleus of the cell. If the structure of this type of cell reminds you of a fortress, that's no accident. Like a castle, the cell appears to be organized to maximize its chances of survival in a hostile world. To visualize this distinction, think about how you might buy apples at the grocery store, for example. Some people just put their apples in their shopping bag, and we'll call them the prokaryotes. Others, like me, are a little bit more meticulous. We place the apples in a clear plastic bag first and tie it closed to prevent any food from leaking onto the apples and to prevent the apples from falling out of the bag. Those shopping bags are like the walls that protect the nucleus in eukaryotic cells. Now, when I tell you what types of organisms are made up of these different categories of cells, the need for this distinction will make more sense. Prokaryotes are mostly bacteria. Why wouldn't bacteria want to protect their DNA? Well, first of all, prokaryotes are thought to be the first living things to have evolved. So they're phylogenetically older than eukaryotes, and the membranes surrounding the nucleus evolved later. And they've also done pretty well without nuclei. They are very well adapted to harsh environments like the snowy surface of Antarctica or the deepest, darkest parts of the ocean. Some prokaryotes don't even need organic compounds to get fuel. They can obtain energy from sources such as hydrogen sulfide, allowing them to survive in conditions where oxygen is scarce, like the very depths of the ocean. We don't fully understand why prokaryotes are so adaptable, but their hardy nature and adaptability has made them the most abundant life form on Earth. The number of prokaryotes in a handful of soil is greater than the number of people who have ever lived on the planet. Bacterial cells are typically much smaller than eukaryotic cells and have a variety of shapes. They are highly successful in part because they can reproduce rapidly when the circumstances are right. Under the best conditions, some species can replicate a whole new generation in under 20 minutes. This rapid rate of replication gives bacteria the opportunity to profit quickly from random mutations. With short generation times and large populations, even rare new mutations can give the species an advantage when it comes to surviving new environments, since they have more opportunities to develop mutations that might protect them from the elements. This is one of the ways in which bacteria can quickly develop a resistance to antibiotic medicines, for example. Another quality that enables antibiotic resistance is the unique mechanisms by which prokaryotes transfer genetic material, or DNA. They can take up genetic material from their surroundings and incorporate it into their own code. Even when the new genes come from a different species, it's as though I had the ability to pick up night vision genes from my cat and insert them into my own DNA. Okay, that's a little creepy. And so enough about bacteria. Let's turn now to eukaryotic cells, the types of cells that our bodies are made up of. Well, those parts of our bodies that aren't bacteria, anyway. Despite the fact that 1 to 3% of our body mass is bacteria, the rest of our bodies consist of eukaryotic animal cells. And let's remember that life has some properties that are common to all organisms. Cells need to sustain and regulate themselves, they need to reproduce, and help the organism grow. All of these functions require cells themselves to be highly organized. A typical animal cell can be thought of as a relatively organized city. It's got a city wall that protects its citizens and keeps out unwanted guests. Inside the cell, we've got a central downtown region or nucleus, where busy executives design, build, and organize the cell's functions. Then there's the hip warehouse district, where the manufacturing happens, and trucks shuttle products from the downtown core to the suburbs and out of the cell entirely. There's a waste management plant that recycles or destroys unwanted byproducts of the cells. And there's an energy plant that fuels the cell and keeps the city humming with activity. Finally, there's the scaffolding throughout the cell that helps the city keep its shape and serves as a network of freeways and side streets to transport proteins and other products throughout the cell. To understand more deeply the different characteristics of a typical eukaryotic cell, let's take an in-depth tour of this microscopic, bustling metropolis. First, let's start by considering what the outer shell of the cell is made of. As we've talked about earlier, the cell's membrane or cell wall is made up of lipids, so it's water-fearing or hydrophobic. This is so that the contents of the cell don't leak out. But the story isn't that simple. If the entire membrane was hydrophobic, then the outside of the cell would repel water, and water carries nutrients to the cell like oxygen and glucose. The cell needs these nutrients to survive. So structurally, lipids resemble jellyfish. They have a spherical head attached to some chains that bring to mind long tentacles. The spherical head is hydrophilic or water-loving, and the tentacles are hydrophobic. So the cell membrane has 2 layers of lipids organized so that the spherical heads are on the outside and the tentacles meet on the inside, like mirror images of each other. This organization then allows the spherical heads that are water-loving to be in contact with the water, either inside or outside of the cell. Scattered throughout the membrane are proteins that act like gates. They help some desirable molecules get across the cell wall and keep the unwanted ones out. Once we get past the cell wall, the next most obvious part of the cell is the nucleus, which as we've talked about, also contains a membrane in eukaryotes. Just like the outer cell wall, the nuclear membrane has a lipid structure, but for added security, the membrane is doubled. That is, there are 2 membranes, each with a lipid bilayer. The wall also has pores or holes that facilitate the passage of certain large proteins and macromolecules. Why the tight security? Because the nucleus contains the blueprint of life, our genetic code, and security breaches could wreak havoc on the functioning and reproduction of the cell, and the larger organism, then, of which it is a part. The main job of the nucleus is to house and protect the cell's genetic information, which is stored in the form of DNA, or deoxyribonucleic acid. Now, as we've learned in the last lecture, proteins are what make a cell function and display the properties of life. And to make proteins, the cell needs to use the DNA blueprint to manufacture and assemble these complex molecules. Inside the nucleus, the all-important job of using the DNA code to generate messenger RNA, or M-ribonucleic acid, occurs. Step 1 in the creation of new proteins. Each gene along a DNA molecule permits the synthesis of specific mRNA, which then interacts with the cell's protein factories to direct the assembly of proteins and their parts. So the DNA creates mRNA in the nucleus, which is then sent out to the protein factories via the pores in the membrane surrounding the nucleus. These protein factories are called ribosomes. Now, ribosomes come in two types: free-floating hippies and unionized working stiffs. Free ribosomes float around in the cell, while the working stiffs, or bound ribosomes, are attached to the endoplasmic reticulum, a network of sacs and tubes that serve as the industrial zone for the cell's various large manufacturing projects, like the synthesis of new membranes or protein gates. Now, don't feel too bad for the working stiffs. They often have a midlife crisis and turn into free-floating hippies. And sometimes the hippies settle down and are bound to the endoplasmic reticulum, or ER. Well, we all have to grow up sometime. All of these different components of the cell underscore the very fundamental characteristics of life: sustenance, reproduction, regulation, and even adaptation. There are three more key parts to the cell worth talking about: the Golgi apparatus, the cell's storage and shipping warehouse; the mitochondria, the energy generator; and the lysosomes, the waste management system. The Golgi apparatus looks like a stack of pancakes. It receives proteins and parts of proteins from the ER and ships them to their desired destination. It's a highly organized system, with one side of the pancake stack receiving parts via transport sacs or vesicles from the ER, and the other side pinching off bits of itself to create new transport vesicles that serve as postage-paid parcels to transport the new proteins to other parts of the cell. Now, the products coming from the ER aren't simply shuttled along. There is some manufacturing that the Golgi apparatus is responsible for. The interior section of the pancakes contains teams of enzymes or proteins that accelerate the speed of chemical reactions, that alter the structure of macromolecules. The Golgi apparatus also acts as a sorting hub by separating proteins in terms of their makeup and making sure that they get to the correct destination elsewhere in the cell. All this manufacturing and shipping requires energy, which is provided by the next structure on our list, the mitochondria. The mitochondria fuel the cell by extracting energy from sugars, fats, and other sources. Cells that require lots of energy to function have many mitochondria, whereas the lazy cells might only have a few. This is how cells are able to sustain themselves and use energy. The mitochondria function by creating ATP, or adenosine triphosphate, which is the energy molecule that fuels the rest of the cell. They create ATP by adding oxygen to sugars, fats, and other compounds. Without oxygen, the mitochondria can't fuel the rest of the cell. This is why we need to breathe and inhale oxygen. There is one other fascinating aspect of the mitochondria that's worth mentioning. Each mitochondrion contains its own DNA, so that the mitochondria can divide and replicate themselves. Now, in sexual animals like humans, mitochondrial DNA is passed down exclusively through the mother. Because it comes only from the mother, mitochondrial DNA doesn't change from parent to offspring the way DNA in the cell nucleus does, which is a combination of genes from two parents. So mitochondrial DNA is often used in genealogical research. Now it's time to turn to the taboo topic of waste management. The lysosome is a sack of enzymes that digests unwanted macromolecules. These can range from sections of damaged cell parts to nefarious bacteria and viruses and other undesirables. Now, if a lysosome bursts and releases its contents into the cell's fluid neutralizes it fairly quickly because the lysosome contents are acidic and the fluid is neutral. But excessive leaking from a large number of lysosomes can be more damaging, and eventually the cell can be destroyed by self-digestion. And so our tour of the cell concludes. Of course, in organisms as complex as humans, there are many different types of cells, from neurons to pancreatic cells to blood cells, and each one has additions or subtractions that support its specific function. The ways that those cell types interact took hundreds of millions of years to evolve. But the cell parts that we've talked about here are common to most, if not all, cells, and serve as the building blocks for our bodies. The organization of a cell seems very complex for such a tiny thing, and yet it's this organization that enables a cell to carry out its many functions, using fuel, replicating, repairing itself, adapting to its surroundings, functions that are at the very heart of our definition of life. But is that where we draw the line? If we take away some small part of a cell, say its genetic material or its ability to fuel itself, but leave everything else intact, is it no longer alive? To answer this question, we need to consider an organism that is at the very boundary of the animate and the inanimate, the frontier of life. The virus. Viruses are worth considering on their own because there is some debate as to whether or not a virus should be considered a living or a non-living thing. What do you think? A virus is essentially a ball of genes packed inside a protein coat called a capsid. It's tiny. Viruses are smaller than bacteria, so small that millions can fit in an area the size of a period at the end of a sentence in 12-point type. But viruses can make us feel terrible when we get a bad cold or even kill us, like the deadly Ebola virus. In fact, the very name virus comes from the Latin root for poison. But viruses can't act alone. A virus can't replicate or power itself. It has no metabolism or ability to generate energy from its environment. It depends on host cells to make new proteins. But it's made up of the molecules of life, DNA, so it can replicate with the help of a host, and therefore it can evolve. These features make classifying viruses as living or non-living difficult. Do they represent the simplest precursors of life, or a complex adaptation that is more akin to a biological machine? And how do we distinguish a biological machine from a living organism? Viruses infect every life form on Earth, from plants to animals to fungi and algae. Until recently, most biologists supposed that viruses evolved after the first living cells and began as bits of discarded cellular and genetic material. But the discovery in 1992 of the large virus, which has many similarities to a small bacterium, has reopened the debate with regards to the origins of viruses. The virus, short for mimicking microbe, has more genetic material than any previously discovered virus. It still lacks ribosomes, so it needs host cells to translate DNA into proteins. But it also has far more translation-associated genes than any previously discovered virus. Is it the missing evolutionary link between viruses and bacteria, or something else altogether? Are viruses in fact precursors of bacteria, or are they simply broken off bits of other cells? We may not be able to answer these questions for some time yet, but there's no question that viruses are well organized and very good at what they do. The complexity of single-celled organisms is remarkable, but in some ways, even more remarkable is the fact that billions of seemingly self-sufficient cells can work together to form complex beings like gorillas, whales, and of course, humans. If we really want to understand why one of the fundamental properties of life is organization, we need to look no further than the tips of our noses. Actually, not even so far as that. We need only to peek inside the most complex thing we've discovered so far in our universe, our brain. Our brain is made up of about 100 billion cells, or neurons. Each of these neurons is connected to other neurons, and information flows from one cell to the others. Some neurons get their information from as many as 10,000 other cells and send it on to thousands more. That's like you receiving 10,000 emails at any given time and simultaneously sending out thousands to your contacts all the time. And that's just one cell. Now, thankfully, most of these emails are pretty simple. They only contain one word, say a yes or a no, a 1 or a 0. The signals that are sent between neurons are binary, just like the code that our computers use. They either encourage the downstream neuron to send its own signal or to keep quiet. But if we look more closely at the junction between one neuron and the next, the story quickly gets far more complicated than that. The space that separates one neuron from another is called a synapse. More specifically, a synapse is the spot where one neuron sends its information and another receives it, like a dock in a shipyard. And just like in a shipyard, the dock is full of action. Ships come in with containers full of different products. Many of those containers are accepted at the dock, and they release their contents into trucks or other vehicles that then transport them to other locations. Some containers can get lost during the transportation. Maybe a crane breaks and the containers are washed back into the sea. Some are rejected by customs because they're suspicious or hold unwanted or contraband goods. And some ships don't even drop off cargo. They just deliver instructions for the customs officials to use. At the synapse, many neurons dock their axons, or the part of the cell that sends out electrical and chemical signals. The receiving neuron, or the destination, also has many docks, or dendrites, to receive the signals. Scattered along the dendrites are customs gates, which analyze the structure of the molecules that are released by the axons of the upstream neurons. Some of those molecules are accepted, and the signal gets propagated down the dock and into the core of the receiving cell. Some are rejected and never make it past the synapse. And some are lost during the journey. They never even make it to the dock. All of these different molecules and their interactions have an effect on the functioning of the cell and its downstream connections. Some molecules are involved in making memories, and they change the way that the neurons get connected. Some turn on or off other functions, like those that cause the cell to fire off a signal or to stop it from firing. Many of our psychiatric disorders result from an imbalance of one type of molecule or one type of gate or receptor. Our pharmacological treatment of diseases like depression, schizophrenia, and anxiety involve replacing missing molecules or altering the gates to make them more or less likely to be accepted. All of this activity, which has profound effects on our psyche, our well-being, and our ability to live, happens in the junction between the two cells. Then, if we move up a level in terms of organization of the brain, we find that there is a pattern to the electrical signals that neurons generate. Certainly, these signals are affected by neurotransmitters, or the chemicals that go across the synapse. But there is also a code in the firing patterns that can be just as important in terms of making memories, or relieving pain, or thinking about physics. We're still sorting out the nuances of the codes, but we know that cells send signals by altering the rates at which they fire, or by firing in bursts, or in synchrony with one another. Each of these codes can be related to different brain functions and malfunctions. The brain's intricate organization can also be seen in its architecture, that is, in the neuroanatomy of different brain regions and the connections between these different parts of the brain. For example, my favorite part of the brain is the hippocampus, because of its intricate organization and architecture, and because it's responsible for creating memories. It's our very own biographer. It's located in what's called the medial temporal lobe. Medial because it's towards the center of the brain, and temporal because it's near the temporal bone, where the temples are located. In fact, the cerebral cortex, or the outermost layer of the brain, is divided up into 4 lobes: frontal, temporal, parietal, and occipital. The frontal lobe is also called the executive, or CEO of the brain. It's the main decision maker, helping us inhibit our impulses and behave in socially appropriate ways. Then the parietal lobe is where much of the information from our senses is integrated, and it helps us focus our attention to things in the world that matter to us. The occipital lobe is largely involved in supporting our ability to see. Seeing is our most important sense, so a lot of the brain is devoted to it. And the temporal lobe is involved in hearing, in understanding language, in storing new memories, and finding meaning in what it is that we're observing. And deep within the temporal lobe is the structurally beautiful and vitally important hippocampus. Without our hippocampus, we can't form new memories. It's the input into the hippocampus that is first affected in patients with Alzheimer's disease. To see the architecture that I find so fascinating, you have to look at a cross-section of the hippocampus. And it looks like a bit like a seahorse, which is where it gets its name, from the Greek hippo, meaning horse, and campos, meaning sea monster. One of the most beautiful images of the organization of the hippocampus was prepared by Santiago Ramón y Cajal, a Spanish neuroscientist often thought of as the father of neuroscience. Cajal is interesting not just for his contributions to neuroscience. He did win a Nobel Prize, after all, but also because he was a skilled artist. His drawings and illustrations continue to populate anatomy textbooks and teach us how our neurons are organized. From these drawings, we can see that brain cells, especially in the hippocampus, but also in other parts of the cortex, are neatly organized into various columns. In fact, this organization is critical for brain function, and if neurons don't travel to where they're supposed to be during the neural development in babies, the consequences for brain function are severe. We'll talk more about the hippocampus later in the course, but for now, let me just point out that its complex neural architecture is what allows us to form new long-term memories. It's what's affected in patients with Alzheimer's disease, as I mentioned, who are stuck in the past because they can't form new memories today. It's what grows larger in London cab drivers when they spend years memorizing the mind-bendingly complex maps of London. It's what we use to recreate events in our lives in vivid detail when we re-experience the past in our mind's eye by remembering. The structure of the hippocampus is a perfect example of why organization matters and why organisms are organized no matter what their level of complexity. Organization equals capability, and capability equals survival. And whatever else life might be for, every example of it that we know of seeks to perpetuate itself. Now it's time to turn our attention to how all of this organization, diversity, and complexity came to be. For that, we need to explore the theory that has completely changed the sciences and is the bedrock upon which modern medicine is built: evolution. In the next lecture, we'll embark on a voyage on the HMS Beagle, the vessel that will take us to the exotic world of Charles Darwin.